We start with the technical session 4 for today, which is for this conference that is conducted by Longevity Asia, Living Longer, Living Better, a symposium on longevity and health. So the first speaker for today's session is Dr. Vivek Tripathi, person who are working on the of NSMCE2 E3 SUMO ligase in replication fork dynamics and chemoresistance. Insights from Indian breast cancer clinical samples and cellular model studies. He is an associate professor at NITTE University Center of Science, Education and Research, NUSCER. So a very warm welcome to Dr. Vivek Tripathi. Please go ahead with you can start with your session.
All right. So first of all, I would say very good morning, everyone. And of course, thanks to the organizers and Chairperson, Professor Gupta, for this opportunity to speak at the Longevity in Asia Forum. So coming straight to the topic without any further loss of time. Today, as we all understand that the threat of this breast cancer across the globe does not require an introduction. And it is estimated, it has been basically statistically, it has been calculated and predicted that this disease will cause the most death than any other malady in developing countries, including India. So therefore, my laboratory is actually trying to understand and develop a simulation-based diagnostic markers for the detection of the breast cancer in the coastal areas of Dakshin Karnataka state in India. So, before I actually take you through the progress, the research progress in my lab, I would like to bring your attention that why exactly we are focusing on this simulation enzyme-based development of these diagnostic markers for the detection of the breast cancer cells. So to clarify this situation, I need to give a brief introduction about the biochemical reactions of this simulation mechanism, which happens within our cells in our body. So actually this, the simulation reaction starts within a cell with the synthesis of a precursor molecule and that gets processed by the SUMO specific proteases to generate basically the diglycine residues at the C-terminal tails of these SUMO molecules. They are actually very small in size, approximately 90 amino acids to 110 amino acids in molecular weight, approximately 8 to 10 kilodalton. They are such a small molecules. And the step which involves the next is that they actually utilizes this ATP hydrolysis and forms a thioester bond with the E1 activating enzyme, which is basically a heterodimeric protein complex. So once this, after the formation of the E1 activating enzyme, this SUMO molecule gets transferred to the E2 enzyme, and then this heterodimeric protein complex is released, and now this SUMO molecule is set to be transferred on the target substrate proteins. So here is the critical step, is that you will be appreciating that only very few E3 SUMO ligases are actually found within our cells, which are actually controlling the modification of thousands of proteins which are involved in multiple cellular processes ranging from DSB repair, single-strand annealing, FOC stabilizer, chromatin structure change, and all that. But my laboratory, here is the point where exactly we are trying to understand the importance of these, a bunch of the E3 SUMO ligases which is found in our cells. So for example, I would like to just throw in a slide where I would like to just show that just malfunctioning or the mutation in one of these E3 SUMO ligases, for example, NSMCE2, can give rise to a spectrum of human carcinomas, including the breast cancer, as you could see here. So this actually, this point actually was the reason where I thought to explore in a detail, because this information which is coming from the TCGA database is still has not been having any clinical implications for the benefits of the cancer patients. So therefore, I to explore the the potential of this enzyme as a diagnostic markers, I collaborated with our hospitals, that is the Justice Case Hegre Charitable Hospital, where the patients are mostly admitted from the rural areas of in neighboring areas of the Mangalore city, as well as some parts of the Kerala as well. So how strategically we try to understand whether NSMCE2 actually can be exploited as a diagnostic marker, since the breast cancer is such a heterogeneous disease. So first thing which we decide that, okay, let's say if any patient is admitted to the hospital, and depending on the tumor size and the other parameters, we decide to collect the biopsies after the consent from the patients. And then the next challenge which comes is that immediately profiling of the nature of these tumor cells, so that we can take a further decision what to do next to save the life of this patient. So what exactly we do, we quickly actually profile whether the tumor is ERPR positive or ERPR negative, depending on the Ki-67 proliferation index. And in the worst case scenario, if all the three receptors like ERPR and HER3 are negative, then that is the TNBC. And sometimes we also go ahead and try to figure out whether the BRCA1 and BRCA2 mutations are also happening in tumor subtypes. So these things are actually approached with, first we go ahead and to find out whether the tumor which this patient is having is actually an invasive state or non-invasive state with the help of H&E staining. And then we further go ahead and do the immunoprofiling or the immunostaining and see the status of these receptors. And once we find whether it is a ductal carcinoma or the lobular carcinoma, we go ahead, depending on the state of the invasive state of the tumor cells, we go ahead and see how far we have to go ahead to collect the biopsies to see what exactly the strategy we can adopt to strategize for the treatment of the cancer patients. So, for example, I would like to just show you recently around a 48 years old female patient was admitted in the hospital. So we just collected the tumor, the clinical samples, tumor as well as the adjacent neighboring tissue sample. And we immediately, after the processing of these tumors, we prepare these paraffin-embedded blocks, and which actually we are trying to generate in a large scale depending on all types of tumor heterogeneity, and which of course would be available for research purposes, whoever is actually interested in that. But the thing is here, so once we prepare these paraffin blocks, we go ahead and perform the immunohistochemical studies first to figure out what exactly is happening or what state of these tumors are present in that the patient. For example, if you see this H&E staining is telling that if you look at this closely, so the bilayer epithelium, actually this is a ductal carcinoma. If you see that the outer layer of the epithelium cell line is broken and the tumor cells have started infiltrating in the basically the global stroma. That is telling that perhaps this stage of the cancer is not, it has reached to the invasive state. So that is why we again try to collect basically the specimens from the lymph node as well to see whether these tumor cells have actually infiltrated or reached to the lymph node. And in that situation, perhaps the treatment strategies might be very harsh, we need to use a very high dose of the chemotherapy, and sometimes that we decide further. But the point which I would like to make here is that how exactly we can use this molecule enzyme, SUMO enzyme, for the detection, how early we can detect these tumor cells by checking the expression of NSMCE2. For example, if you would say, we also took a small piece of these specimens, and after the purification of total RNA, we synthesized the cDNA and performed the QRT-PCR, and you could see that approximately 4 to 5-fold increase we can clearly see in the expression of NSMCE2. So now the next challenge is that we have to find whether we can also, in a non-invasive state, whether it's a ductal or the lobular carcinoma, we can also see that kind of increase, which can actually help us to detect these tumor cells, and it is going to strategize our therapeutic strategies to save the lives of the cancer suffering patients. So, the thing here is that this is not only the strategy which we are having once we find the expression of NSMCE2, but how exactly we can try to inhibit the activity of this E3 enzyme. That is another challenge which I'm doing. So since in India, lots of jadi booties are used, for example, the phytochemicals and all that. So we have a list of the library in our pharmacy department, which I'm still collaborating, and trying to screen that how the two important residues which are present in the sp ring domain of this NSMCE2, that is the cysteine 185 and the histidine 187. So we are trying to screen if we can effectively block these amino acids to inhibit this activity, so that we can try to regress and test for further validation. So that is another step which we have been taking forward. So that is not only basically the focus of my lab. In parallel, we have been also trying to perform a basic research in the lab after getting a very intensive research exposures from during my PhD work from the IGBMC University of Strasbourg, France. And then I also from the lab of Dr. Sagar and Dr. Arnav. Here, what exactly the angle which I'm trying to understand at the molecular level is that the NSMCE2's role in the replication fork dynamics. Because sometimes what happens in the TNBC cells, due to the absence of all three types of receptors, they are very difficult to treat. So therefore, if we can somehow modulate the replication machinery, then perhaps we can develop assimilation-based therapeutic targets. So this is absolutely a new field where it's a very growing field where it has been found that upon the replication stress, we have a very specialized set of proteins that is called as the translocases, like SMARCAL1, ZRAND3, and HLTF. They are the translocases, and upon the replication stress, the actively growing replication forks gets reversed by the annealing of these newly synthesized daughter strands accompanied with the re-annealing of the parental strands. And the protection and the efficient restart of these reverse forks are paramount importance and for the maintenance of genomic stability. So the role of NSMCE2 is not very well known here. But what all we understand here is that NSMCE2 is actually, with the help of other fork-associated factors like SMC5 and 6, it is involved in the efficient fork restart and somehow promoting the growth of the tumor cells. So that is still in investigation. We will try to take it forward. So we have been also using a highly sophisticated technique like a DNA fiber assay to understand how, if we apply our experimental conditions, and how exactly these novel inhibitors of the NSMCE2 can really be exploited at the cellular level to test whether it's really effective to consider for take it forward for further preclinical studies. So DNA fiber assay is another very quickly. I would like to just, since we don't have much time, so I'll just quickly in a minute or so, I'll just explain about this technique. So this is a very interesting technique, which at the single molecular level, we can track the speed of the the movement of replication forks, where we can do a sequential pulse labeling of the actively growing replication forks with the this thymidine analog CNDU and IDU, and then with the chemical process, like we can spread these DNA fibers on a 45-degree angle, followed by we can perform the immunostaining, and we can detect the length of DNA fiber spreads, as you could see here. So these are actually the individual replication forks, and this is telling the labeling of the first label CLDU and IDU, and that basically is telling, and depending on the statistical analysis, we can calculate the movement of individual replication forks in micrometer per minute. So these are the different types of experimental conditions which we'll be applying to investigate how effectively we can prove whether the NSMCE2 can really be targeted for such kind of, not only for the identification of the breast cancer in early stages, as well as at the molecular level, where exactly NSMCE2 is having its involvement in the modulation of replication fork dynamics. So I think I will stop here since we do not have much time. So in acknowledgements, I would like to say I have a bunch of people in my laboratory, very talented people. And of course, I would like to acknowledge the collaborators, Professor Dr. Anirban Chakravarti, who is now director of the institute, and Dr. Vinay from the hospital, and Dr. Praveen from the pharmacy department. And currently, I do have a financial support from the university seed money to start my lab. And of course, I'm interested to get more external fundings in near future. Hopefully, we'll do more exciting work and share with you guys. Thank you very much for your attention. Thank you.